close. Sorry. Streamlabs. It could also be the Streamlabs is being a little finicky today as well. So hopefully the stream will go well today. I really hope so. But yes, welcome everybody. Welcome. If you have any questions, as always, place them in the chat and I will answer them if I can. As you notice, I've got closed captioning going. Yay! I'm so excited. Finally got that to work and I had to do some finicky with that. I'm learning every day better ways to make stuff work on my stream, especially with audio using a Mac. So let me know if the music is too loud or if I sound a little off. I was doing test recordings and they seem to be okay on my end, but you never know when it's stream how they sound. But yes, so just a recap of what we have done so far. So the beginning of this whole cloning procedure, and here I'll post up cloning. Let's see, cloning. Oh my goodness. Yeah, closed caption's gonna be fun. If you want to clip the fun closed captions, that would be great. I would love it. I love closed caption fails. But yes, so we are finishing basically the cloning procedure. We only have one more thing left to do for this whole thing. So we started off by constructing our plasmid, our double-stranded DNA. We transformed it into Eli Ecoli. We picked Eli Ecoli off of plates, and then we grew them up into our tubes right here. So here are our tubes. They are all ready to go, and we are going to extract our plasmid DNA out of Eli Ecoli today. So that is the plan. It's going to take about an hour or so for the actual protocol, but I'm going to stick around so we can chat some more, get more people involved if we can. And yes, also let me know if the captions need to be bigger as well, if they're easier to read. And all of these, if you've missed the past streams, you can catch them on YouTube. There is my YouTube channel for the VODs, and of course Twitch keeps the VODs for 2 weeks up on my channel here in Twitch, so they should be all set. But yeah, so plasmid extraction or plasmid preps, as I will probably we'll probably switch between now. I'll probably say plasmid preps more. Plasmid traps. Oh no, closed captions with science. It's gonna be fun. Plasmid preps. There we go. Plasmid wraps and preps. It works. Plasmid preps. Basically just means that we're going to lyse poor Eli Ecoli. We're going to break holes in his little membrane and get the DNA that's been holed up inside of him today. So we're not going to be nice to Eli today, unfortunately, but we're okay. We're okay. There's lots of Eli Ecoli around. So we're going to basically go from this to this while we do plasmid preps. And I mentioned it before in my previous stream. How we do this experiment is we use a kit. So it's basically something that comes with all the buffers that we're going to need. So the buffer that we use to basically spin these guys down and resuspend them in a buffer. Then the lysing buffer. So lysing means that we're going to extract all of the stuff out of him. And then we'll stop that because we don't want it going too long, or else it'll destroy the DNA. So there's an agent to stop that, a buffer to stop that. Then we will spin it into a column, which has a membrane attached to it, which will capture the nucleic acids, in this case DNA. And then we'll wash it, and we'll take the DNA off of the membrane using water, and we'll check the concentration. So how much DNA we basically got from Eli Ecoli today. So that is the plan. That's the protocol in a nutshell. Very quick way to go through it. And we can get started. And if you have any questions, then I will get to them as I see it. So welcome to the newcomers in chat. glad you could join me today, and I hope we have a good stream and no more hiccups. Hopefully it's all working good. Alright, so why don't we get started? I'm going to just start prepping basically. First thing we have to do, PPE. I already got my first set of PPE on. I've got my glasses, my safety glasses. And I actually need to clean them off because I've got a bunch of fingerprints on them. So I'm going to clean them off real quick. But yes, let me know if the music is too loud. Like I said, I was messing around with the audio yesterday after I broke it. Then I fixed it. So if the levels need to be changed up, let me know. I can definitely do that. Move up my microphone a little bit. It fell down just a little bit. Alright, so we're going to say goodbye to our mascots as I get prepped for doing our plasmid preps today. Next PP that we don is the lab coat. And my favorite lab coat, of course, is purple. Just snap it on. There we go. Purple lab coat, purple lab coat. And you know what? We've listened to this song a couple times. Let's see if I can go to some different ones. Let's go to the next one. Let's go to the next one. There we go. Sorry, just wanted to change it up before I put my gloves on. Let's get gloves on, so that's the next step of PPE. Alright, don the gloves. And then we will be all set and ready to go. Alright, so got the PPE on. We're all set. We're good for safety. So here are my tubes. So if you remember, these are the tubes that we were growing up E. coli in. So they're nice and cloudy, meaning that we've got E. coli in there. And I already have to tell you, welcome to the first step of science where stuff doesn't work. Here is A and B. Unfortunately, it's clear, so it means that they did not grow up. So this happens every once in a while. It is not a surprise to me sometimes, where the E. coli looks like it's good on the plates, but as soon as you put it in the liquid media, it will not grow. And it's not because it's the concentration of the antibiotic, it's just sometimes we get some contamination that happens on the plates. Looks like what we want it to be. It probably was just like something pretending to be E. coli that I picked. And I mean, it happens all the time. So unfortunately, A and B, we got nothing. And this is just to show you what it looks like. So here's C and D, nice and cloudy. A and B, there's nothing there. We've had this every once in a while in the lab. It happens. But it just means that instead of 10 samples, we're going to be doing 8, and I'll have to redo the ligation for A and B. Time to troubleshoot that one. So I'm going to go throw these away since there's nothing in here. Throw them away in our biohaz. So I'll just have to go back to the drawing board with A and B. So we're just going to be doing C and D, so 8 samples today. And this one requires a little bit more starting prep as far as... Whoops, I almost dropped my tips. What we're going to be doing today. So we're going to be using mainly our P1000, so the one that goes up to 1,000 microliters or 1 mil. We're going to mainly be using this. And the 200, so we're doing larger volumes today. I'm going to move these out of the way. And then I'm going to turn on our centrifuge. So the thing that spins these guys really, really fast. And it's a little loud. That's all right. But what I need to do is label. I have to label all the tubes that we're going to be using. So we're basically, these are the tubes that I'm going to use to isolate the actual E. coli cells that are in this liquid media that we have. So I need to spin these down. Right now they're within the media. We don't want the media when we're doing the extraction because the salts and everything in here will be a problem in the actual DNA isolation. So we spin it down. We will spin these down, gather the cells, and move forward with the extraction. So that's what these set of tubes are going to be. These sets are going to be when I actually take the DNA off of the column, and I will show you what the columns look like. I'm gonna move that there. So these are what the columns look like. So let's pull one out for you. So this is what it looks like. So it's basically a tube, and this is the column. And so that white thing right there, let's see if I can pull it down. White thing at the bottom is where the membrane is. This is what's going to capture our nucleic acids. So right there. That's where the DNA is going to sit. So I need a column for each of these. And as you can see, we just buy these. They're part of a kit as well. So now it's just labeling everything. I'm going to also grab the buffers that I need over here and put them off to the side. Have it there. And then grab some extra paper towels. So I'm going to show you how I do the extractions, how I do the preps. Other labs do it in different ways. This is just how I do it. But all I'm going to do right now is I'm going to label the tubes, and we'll chill out to some music. All right, so I got the labeling done. All the tubes are set. All I did, since we're only using C and D today, I just labeled 1 through 8 and then put C and D on the final tubes in the back. All right, so the first thing, I'm going to change our volume. Let's see, I know this got a little bit of a, there we go, to 750 microliters. And we're using the filter tips today. I don't want the E. coli to be going into my pipette. And I'm going to pipette 1.5 mils of our E. coli into these tubes. And that's just because that's how much the tubes will hold. So two 750s. And then I'm not going to use all of it because I actually, if these end up being what I want them to be, I will make stocks of these. So I will actually use these so that we can keep them for later if we want to use them for other experiments in the future. So I'm just going to do this for each of them. Obviously making sure that I'm putting them in the right tube. I've done that many times before, where I'm not paying enough attention and I put them in the wrong tube. The good thing is you can easily change the number on the tubes if you do that, because it happens. It happens. We'll just do this for all of them. So just moving liquids from one tube to another, our favorite thing to do. At least it's not colorless. We get to have a nice little yellow color this time. But yes, one of our many things we do in the lab, right? 3 things we do in the lab. We move colorless liquids from one tube to another, from one place to another, wait for centrifuges to stop spinning, and wait for things to thaw. So today we get to do the wait for centrifuge to stop spinning as well today. So, get to join me for the funness that is that. And I'm definitely switching tips between these because we do not want to get cross-contamination. We want to keep these separate, no mixing, because one of these, remember, one of these comes from a single colony on the plate, and that colony came from one Eli Ecoli cell that picked up our plasmid. And we just want to make sure nothing funky happens just because we have more than one Eli Ecoli cell in here. Alright, one last one. One last one, and then we'll be all set. So the next thing I'll do is I will spin these in the centrifuge for a minute at 18,000 times G. Basically, that means 18,000 times gravity, times the force of gravity. It's fast. It's extremely fast. So I'm going to put these in the centrifuge. I will be right back. Oh no. Turn it back on. And while I'm doing that, I'm going to put these in the fridge. We actually keep these in the fridge until we're done checking them. So we'll do the restriction enzyme digest to check these next week. And we'll go from there. All right, here we go. All right, so I'm just putting these in the centrifuge. Real quick. And then we'll get this spinning. You might be able to hear it. So that's just me putting the top on the rotor. And these are spinning. All right, so we're gonna keep this here. My little waste beaker is right here because we want to get rid of the liquid media. So this is a waste beaker. That's where we put the waste in here. And then I'm going to grab our first 3 buffers. The other one that we have is in the fridge. So I'm gonna grab the one in the fridge. And so the first buffer we're gonna use, this is buffer P1. This is what we use to resuspend the cells after we get them out of the liquid media. Buffer P2, which is blue. You can see it, it's blue. Nice blue color. This is the lysis buffer. So this is what's going to put the holes in our E. coli and cause it to spill out all of its cellular components into the tube. Then we have buffer N3, so this is neutralization buffer. This is what's going to stop the lysis from happening so that we don't destroy our DNA. All right, and that should almost be done. So I'm going to move that here. And I'm going to change this. So if we're using the buffers, hard to see there. There we go. We set it at 250 microliters. There we go. And I'm going to grab some more tips. As I see, I only have a couple of E200 filter tips. I can open these. There we go. So we have those ready to go. All right, so they're done spinning. I'm going to take them out of the centrifuge. So these are now all the cells, all the E. coli cells have been collected at the bottom of the tube because we spun them so fast. And the liquid media is going to be throughout the rest of it, and I will show you in 1 second what they look like. All right. And then I will also show you why I have paper towels right here. All right, so this is our tube right here. So this is one of the tubes. Actually, it might be easier to see. You can see that. So all of the E. coli cells are now in this pellet at the bottom, and the rest of this is the liquid media. So we isolated all the Eli Ecoli cells that we transformed here, right at the bottom. So now, what I do, this is my trick. This is my trick. Not everybody does this. This is just what I do. To get rid of liquid media, we dump it off, and the pellet's still there. Then I do this. So this is my trick. I'm just sharing my trick on how I do this. because we want to get as much liquid media off as possible so it doesn't interfere with the downstream extraction process. So if you've seen people do this before, which you might have, or you do this in your own lab, this is just the way I do it. There's no right or wrong way to do it. I think the wrong way would be if you decide to basically mix it all back up again. That would be the wrong way. You don't want to do it that way. That defeat the purpose of what you just did. And then I tap it until I don't see any liquid coming off it anymore, just like that. And then we just have our pellets. So just a little tap, until the liquid's gone out too. So just like that. It's effective, it works. And the good thing is it's easy cleanup as well. Because after this is done, I can just get rid of the paper towels and we'll spray it down with ethanol to clean it up. And also I know that I spun these in microcentrifuge tubes, so these are microcentrifuge tubes. Other people, other labs spin them right in the culture tubes. I just like having it in, to me, it's just more convenient putting in the microcentrifuge tubes. And I'm just gonna spray around. Wipe it off. Where we were working. Just get that clean with 70% ethanol. There. Just in case there was we were putting the media on there just in case. I like to keep it nice and clean. Alright, so we are going to add the P1 buffer. So this will resuspend the cells in, and I'm going to use one tip. This is tips and tricks. Let's see if I can move so you can see what I'm doing. Just shooting it directly into the tube, so I don't have to change tips. Save on plastics. Filter tips are expensive, and we use them a lot. So this is a way to save, and if you're good at it. So obviously, if you accidentally touch the tube side, you definitely want to change the tip. If you want to directly pipette them into the tube, change the tip. You don't want to contaminate at this point. Alright, so we're all done with that. And then to resuspend them, I like to use my P200. So now we're just going to pipette up and down to resuspend the pellet so they get back into solution until I don't see any more chunks in there anymore. There we go. So it turns into a cloudy. It turns cloudy when you're done with that. And the pellet's gone. So we just do this for all of them. I'm moving a little farther off so that you guys can see it better. See what I'm doing. All I'm doing to mix it up, and I'm closing them because I want to make sure that I'm not going back into one I've already done, is I'm just pipetting up and down. Up and down. And I'm using a filter tip because just in case it goes up there and I just touched it, that's okay. Just in case the E. coli somehow gets up towards the filter, I don't want it to get into my pipette and contaminate my pipette. And this just helps me keep track of which ones I've done so far. Hopefully you guys can see it. I know it's a little hard to see when I'm working this small. There we go. So this will get these suspended in the correct buffer for doing the actual extraction. That's why we're doing this right now. And that's just the way this kit works. Most kits have this step because you want them to be suspended in a solution. It makes the extraction process easier. I think another kit you don't have to, and you just add the lysis buffer and then you suspend in the lysis buffer. But we use this kit just because it's readily available to us. But yes, if you have any questions, please feel free to put it in chat and I can answer them for you. But yes, I'm hoping that you have a wonderful day. all the new people that have appeared in chat, welcome to the bench. Today we're doing plasmid DNA extractions from our E. coli. If you want to know what E. coli is, there is a command for that, exclamation point E. coli. But this is part of the cloning process that I've done over the past couple of streams, and we're just continuing it today. Alright, I'm going to open these back up. They're all thoroughly resuspended. And now we're going to add the P2, which is, buffer P2, which is our lysis buffer. So, say goodbye to Eli Ecoli. Bye, Eli Ecoli. We're still using 250 microliters. This is what's going to lyse our E. coli. They're going to end up getting nice and lysed, just in case I had a bubble that touched the side. I want to make sure that it's being extra cautious. And it will give us the DNA that is within the cells. So you want to act pretty quickly on this. You don't want it to sit in the lysis buffer for more than 2 minutes. After 2 minutes, it will start to degrade your DNA. So this is why I know what my limit is. This is essentially the rate limiting step. If you have too many samples, they will end up sitting in the lysis buffer for longer than 2 minutes, and then that's just bad. So you don't want that. So I'm not mixing with the pipette because this is better to mix this way. It makes it better. It will get it nice and lysed this way. The pipette can be a little too harsh. We don't want to get too much going, and just do this a couple times. And they're ready to go. And now we're going to stop it. And we're going to change this to 350 microliters. And this will neutralize the lysis, so it'll stop the lysis. So it will preserve the DNA. But at this point, our Eli Ecoli is now gone. They are no more. The lysis is extremely harsh. They have become cell components at this point. Bye, Eli Ecoli. I'm so sorry. Alright. And now, just close them up. Do the same thing. We do this. Mix it up. Same with these guys, and it has sufficiently stopped the reaction. And a good way to know is you end up getting this white cloudy spots in here. So if you can see it, there's a white cloudy spot. So that is basically aggregating any proteins, any lipids that are in there, so fatty molecules from the cell membrane, and the nucleic acids should be sitting in the clear liquid. We're going to spin these down so that we can actually collect the clear liquid from this, and that is where our DNA is going to be sitting. So these are going to spin for 10 minutes at the same speed, so 18,000 times G, and that will help drive all the cellular components. So like I said, the lipids and proteins, they will actually collect on the side, allowing us to get the clear liquid where our nucleic acids are sitting onto the column. So that's when we're going to use the column. So while we're waiting for that, I'm going to put our buffers away since we don't need these anymore, and I like to keep a clean bench as I'm working. So I'm going to do that real quick. And they just go where we keep all our kits. I'm also going to get rid of our empty tip box. So we recycle these, which is nice. So at least we try to help the environment by recycling these. All right. Now, while these are spinning, we're going to be using 2 buffers after this to clean up the DNA on the column. We're using buffer PB, which helps to bind the nucleic acids to the membrane. I mean, it'll work just fine without this, but this is like essentially an extra cleaning step. And then buffer PE, which includes ethanol, to clean it up. So it'll get rid of any contaminating salts that could be in there from the leftover buffer. If I didn't immediately get rid of the liquid media completely, that'll help get rid of that as well. So we'll use these to clean these up once they're on the column, once the DNA is on the column. And I'm just going to get everything set for that time. First step, obviously, is to move the liquid from the tubes into the columns. So that'll be the first thing that we do. And then I'm going to be using this after we're done extracting to check the concentration of DNA, so I'm just going to have that sit right there. I'm going to take off my gloves for right now, just so I can check on chat and see how you guys are going. Welcome the new people that have arrived in chat. Yes, welcome, welcome. We're sitting at the bench doing our plasmid preps today. So we got rid of our poor Eli Ecoli. Poor Eli Ecoli. This poor guy. We had to put some holes in his membrane in order to get the plasmid DNA. That's alright. At least we still have this cute, adorable mascot for us. But yeah, so this is the longest part of the protocol. We're just sitting in waiting for a centrifuge to stop spinning. Like I said, one of the 3 things most scientists do at the bench is wait for a centrifuge to stop spinning, moving clear liquids from one place to another, and waiting for things to thaw. So 2 of the 3 today. 2 of the 3 today ain't bad. I'm enjoying these closed captions. I really am. This is great. If you guys want to clip any of this stuff, they're awesome. Awesome, and share. I love it. You can join our Discord. Oop, it would help if I was in actual chat. And you can post the clips there. That would be amazing if you would like to do that. So feel free to join our Discord. You can keep up with the channel outside of the streams. I also post when I'm going live, also on my Twitter. The VODs will be up on the YouTube channel. I'm gonna shameless plug YouTube channel for those who missed it earlier. So if you missed a previous recording, you can see it there. The VODs are also going to be up on Twitch as well. So if you would like to see this from the very beginning, where we constructed our plasmid, so we constructed our double-stranded DNA, then we transformed our DNA into Eli Ecoli. We picked our Eli Ecoli off of plates, grew them up, and today we're extracting the plasmid DNA from them, doing plasmid preps. So this is a typical cloning technique that many labs use. It's hugely used if all you're doing is plasmids. And you can even go, so I'm doing small preps, but you can actually do preps from Erlenmeyer flasks. And those are usually called maxi preps in that case, because you're talking like 50 mils or 100 mils of E. coli that you grew up, or bacteria that you grew up. It was a lot. We don't do that here. We don't have to grow up that much. We don't need to do that. We just do small ones. But yeah, so I hope you're having a good day. It's afternoon for me today. It's already noon today. But if it's morning or evening, I hope you're having a good day either way on this lovely Wednesday. And you're enjoying the vibes of our stream today. I know the beginning of it was a little shaky. Had some issues with the audio. That's why I didn't stream yesterday, in case you missed the beginning of that. I unfortunately broke my audio yesterday, so it was a lot of troubleshooting. It was just troubleshooting all day for me, but I fixed it, and then we just had a slight snafu when I first went live. I had to restart Streamlabs because the web camera was having some issues and lost one of my media streams, but seems to be going good right now. And the closed captions are available now, so I hope this is helping in case you would rather have closed captions instead of listening to me if you have it on mute. I know it's not picking up everything that I'm saying correctly, but hopefully I get the gist of it. But yeah, so if you have any questions, please put them in chat. I will gladly answer questions. You're just going to be hanging out today doing these plasmid preps. So this is basically halfway. We are halfway through the procedure. We'll just grab our DNA from the liquids, clean it up, check the concentration, see how much we've got to go with for doing restriction enzyme digest, and that I'll be doing that next week. I won't be able to stream the rest of the week. I'm extremely busy doing other things outside of stream. But next week I plan on doing the restriction digest. That's where we run bands of DNA out on a gel. The enzyme's going to cut in specific spots in the DNA, in the plasmid DNA, and we match up the bands that run on a gel to what we expect if we had the transformation go correctly, if we constructed our plasmid correctly. So here's hoping that's what we get. I want to see beautiful bands that match up to what I expect. So I haven't decided when I'll be doing that next week. It might be Tuesday, because Monday I will be busy. But I will let you know. I will let you know if you want to check my schedule on Twitch. That's where I will put when I'm going to be streaming. I'll also change the title so you have an idea of what we'll actually be doing in that time frame. And if you would like to follow and hit that notification bell, then you will know from Twitch. Twitch will give you a notification when I'm live next as well. And I also post on my Twitter when I will be going live, and of course in the Discord channel. So if you would like to join Discord, that would be great. That would be awesome. Talk science, talk other stuff. You don't have to talk science all the time. We have a general chat. We have a gaming chat. So if you like to talk some games, you can do that too. But yes, welcome. Let's see, where are we at? We have 2 minutes. 2 minutes left on the centrifuge. 2 minutes left on the centrifuge, and we'll be ready to move on with the procedure. So, awesome. Yay, I'm glad you could hang out with me today. And I noticed that Castro, you were in chat. I think you missed us getting rid of poor Eli Ecoli. Eli Ecoli is no longer available. He has now been reduced to cellular components. So, I am sorry. It is what it is. But yes, I hope the audio is better for everyone today, and we don't get as many of the popping noise that was showing up in other streams. If it's still there, it seems to be yes, sad kitty Castro. I know. Bye Eli. It seems to be a Mac issue, unfortunately. So if the pops are there, I unfortunately can't get rid of them. I apologize. They can be annoying, I know. But hopefully it's not as bad. It's not as bad and annoying as they were before. And I hope the music is okay. Like I said, I had to mess around with the audio a lot yesterday. So I missed all of my settings that I had before got mixed up. So it sounded good on recordings. Hopefully it sounds good in stream as well. It's still there a little bit? Yeah. Oh well. I tried. I looked up everything I could, did some deep dives, but it seems to be an issue with actual Macs. They fix it, and then it breaks again, and they fix it, and then it breaks again. Unfortunately, we're in the breaks again issue with it, so I just hope it's not as bad. And you can hear me and the music better, and not so much the popping anymore. But that's okay. We'll get through it. Oh, just heard the centrifuge. Centrifuge is done. All right, let's put on our gloves and go grab our samples out of the centrifuge. That was the longest centrifuge step, longest spin. That was 10 minutes. The rest of the spins are going to be only 4 minutes. So we have a couple more. All right. And then I can show you the results of this centrifuge step. All right, so here they are. And hopefully you can see. So we've got this white stuff right here. These are cellular components. So this is going to be the lipids, so the membrane pieces. The proteins, they're all right here. And we've got the nice clear liquid, and that should be where our nucleic acids are sitting. So what I'm going to do is I have this set at 800 microliters, and that's just because that's the max volume you can put in the columns. I'm going to carefully pipette, and you can probably see this better on the black. I'm going to carefully pipette. There we go. Thankfully, you can work around it if you get the little chunkies. You just don't want a lot of the little chunkies. Carefully pipette the clear air, and then stick it in the column, just like that. Now we're all done. We don't want any of this in the tube, so we actually throw it away. So we're all done. So I'm just going to do this for all of the samples. As you can see, I'm not taking every single bit of the liquid, so there is some still left in the bottom. I don't know if you guys can see it, but there's a little bit left. I don't want to take that because I don't want to get too close to the white cell components, because then it's going to clog up the membrane and we won't be able to get clean DNA off of the membrane when we elute. It will just take up space where the DNA is supposed to be in the membrane, and any salts that are attached to that are just going to make it not clean as well. Because salts in downstream, so the restriction enzymes can cause the actual enzyme, so the protein that works, to not be as efficient or not work at all. So we want to get rid of that, and that's what the cleaning steps are for. But if there's the white stuff on it, it's not going to clean it very well. So that's why I'm leaving a little bit of the liquid in the tube. There. I'm also making sure that I pay attention to what column I'm putting it in. Like I said before, I have done it many times where I'm not paying attention and I put the wrong sample in the wrong tube, and thankfully, once again, we can always just relabel, which is good. But the best way to avoid that is to pay attention to what you're doing in the first place. So I'm being very careful to watch what I'm doing. Great. All right. We're all set. Oh, I'm going to have to get some more. All right. So now that we have that set, we want to get rid of all the liquid here because there could be some salts left over from this. Right now this is a dirty prep. So we need to get rid of the liquid. I'm going to spin this in the centrifuge for a minute at 18,000 times G. So we don't change the speed throughout this entire protocol. It stays the same, which is great, because then you don't have to worry about remembering to change the speed. All you have to do is change the time for the one long spin, and everything else is just a minute at the exact same speed. All right. I'm going to grab some more filter tips, because we need more filter tips. Get these all set and ready. There we go. And change this to, there we go, 500 microliters. So this, we're going to use 500 microliters for our first cleaning step, which is buffer PB. It'll help the DNA bind, and it's an extra step. So like I said, you don't have to use this buffer. It just helps. It's really good if you want to have really clean DNA at the end of the prep, as well as an extra step to just get rid of any other contamination that can be showing up. So we always do this step. It is optional. If you look at the actual protocol, it says it's optional. This is what we use. We like to continue this in our lab. I like to have nice clean DNA, because if I don't know what we might be using it downstream. I know we're definitely doing the restriction enzymes, but if I want to send it for sequencing to double check what the actual DNA sequence of our plasmids are, it helps that it's really clean DNA. All right, and it's done spinning. Let's grab these guys. Alright. Okay. So here we have them, and I will show you. So we don't have the liquid in the top anymore. You can see it's all flown through the actual column into the bottom of the tube. So what should be on the membrane right now is our nucleic acids. And we don't need it's called flow through or supernatant. That's what the liquid that goes through the column is called. We don't need that, so we're actually going to just dump it in our liquid waste. You can do 2 at a time. If you're confident, you can do 2 at a time. Makes it go a little bit faster. Alright. Okay. So now we're going to add 500 microliters of the buffer to columns. Trying to angle it so you guys can see what I'm doing. And once again, I'm using the same tip just to help reduce the amount of tip waste that we have in the lab. Really a little bit environment friendly. We go through a lot of plastics, unfortunately, in the lab. I try to do my part. Like recycling the tip boxes when we're all done. And thankfully, our university lets us do that. Because some places you're not able to do that. So before I get this spinning, I'm actually going to change this to our final. We're going to go right back to 750 microliters for the final step. All right, now we're going to centrifuge these guys for another minute. And so this buffer is going to help the DNA bind to the membrane. It will also help clean it up as the solution goes through the column. Nice clean DNA. There. So this goes, I'm going to go put this back. We don't need it anymore. Like I said, I like to try to keep my bench nice and clean. And the buffer's got a little bit of white stuff from the buffer on my bench. Get rid of that. Ooh, and I'm going to go put the empty tip box away. I'll be right back. Alright, I'm back. Okay, so this is going to be our final cleaning step right here. This is buffer PE. This has ethanol in it. And ethanol is a great wash for DNA. You see it a lot in other extractions as well. So we'll do this one. And then after the washes, we are going to elute the DNA. So we're going to pull the DNA off of the membrane. And then we'll check the concentration and see how much DNA we were able to extract from our E. coli. Oh, it's done. Let's grab these guys. All right, so similar to the first spin that we did, there's just less volume. The buffer went through, collected at bottom. So we have our first cleaning step done. And we just get rid of it. We don't need that. Throw it away, essentially, in our liquid waste. There we go. And now we'll add the final buffer to our buffer PE. Oop, nope. Alright, so I just did that, and I should have showed it to you as to why. I had a bubble in the middle of my pipette, and that's going to be inaccurate as far as the measurement goes. So that's why I repipetted the buffer. And that's just because the solution is very... oh gosh, the opposite of viscous. Why can't I think of the opposite of viscous? Oh goodness, I can't think of the word right now. Opposite of viscous. Opposite of viscous. It likes to flow out of the tube. And it does it easily. So that's why I'm also going a little bit fast with it. or else it'll drip everywhere and make a mess. Gosh, I can't think of the opposite of viscous today. All right, so this is our last spin. Well, not the last spin, sorry. The opposite of biscuits. I saw that. Think of biscuits. Oh, goodness. Closed caption, I'm not saying biscuits, except then. So this is going to be our last wash step, and we will centrifuge these for a minute. And get ethanol wash done. So after this, we're going to do an extra spin to get rid of any residual ethanol, so any ethanol that might be sitting still in the column. If that stays there, it can mess with downstream, so it can have effect on downstream. So with the restriction enzyme digest, the ethanol, those enzymes don't like to have ethanol in there, so it'll reduce their activity, or it could completely get rid of their activity. So we do an extra spin after we do the wash, just to make sure any of the ethanol is completely gone from the column. And let's get... I'm gonna get another pipette just because it's easier. Not that one. There we go. I'm just getting another pipette just because it's easier. We're going to add 31 microliters of water to the column after the washes. And that's what we're going to elute in. So we'll add the water to the column. It's going to sit for a minute on the column. So I'm going to get this set up right here for a minute. We'll need our handy dandy timer. I'm going to put, we do not need our P1000s anymore. Oop, it's done. It'll sit for a minute and then we spin it down and the water will go through and take all of the DNA off of the column. And the DNA will be in the water at the end. All right, so once again, the ethanol wash has gone through. We're going to get rid of it since we don't need it. And then we'll do the extra spin to get rid of any ethanol that is sitting on the column, so it doesn't disturb our downstream processes, so it won't affect the digest that we're going to be doing next week. All right, so it spins another minute, and that'll collect the rest of the ethanol. There. All right, so that's going. All done with liquid waste. I will get rid of that off stream. We just have a chemical container that we put this in because it does include ethanol and the detergent for the lysis, which you cannot put down the drain. No, that is a hazardous chemical. You don't want to put that down the drain. So we actually collect this into containers that will eventually go to our chemical waste department. So our environmental services will take care of that first and dispose of it correctly. So another safety tip from the lab. No hazardous chemicals down the drain, please. That is bad. Let's not affect our water ecosystem by putting that down the drain. It would be very, very bad. We don't want to do that. All right, and that's almost done. Are there any questions that you guys might have about the process if you just came in and you're like, what exactly is this thing that you're doing? Feel free to put it in chat. That'll be great. I will answer your questions for you. If it's a question of another science question, I can also try to answer it. If I don't know the answer, I will look it up and get back to you about it. Not a problem. Not a problem. All right. And I can show you. So there is definitely ethanol still left on it, and I can show you. See? So there is still ethanol on there. So we're going to get rid of this ethanol, if you can see it. There's still some. There you go. There's a water in there. Not water, but it's ethanol. So that's why we did it. So the next step, because we don't need to do another wash, we're going to take the column out and put it directly into these tubes. So these tubes are our final tubes. These are where we're going to have the DNA go through with the water. And And then I'm just going to throw these guys away. We don't need them anymore. All right. All right. So looks slightly different. We're putting in the microcentrifuge tubes. Now we're going to add 31 microliters of water to each of the columns. Why am I using 31? Because I'm going to use a microliter of this water at the end. All right, this might be, I don't know if you guys are going to be able to see this well. I'm going to put the water right on top of that membrane, like right on top of it. I don't want it on the sides, I want it directly on the membrane because that's where all the nucleic acids are, and if it's on the side, it won't be able to make that good contact with the membrane and get all the DNA off. And I am changing tips, just in case I touch that membrane. I don't want to touch the membrane because it's fragile, so you sit it right above it. You don't want to puncture through it, that would be bad. Then you're just going to have the water just go through because you punctured it, and you won't collect any of your DNA. So I'm just doing it right on top, trying to get it right in the middle of that membrane as well. So it's not sticking on the sides, it's just good practice that way. You want to collect as much DNA off the membrane as you can. And I'm running out of water in my little thing. Hopefully I've got enough. Probably going to come right down to the last one. Oh, nope, not enough. All right, I need to get some more water, which is easy. I have my own little separate thing of water. I just keep it in it. So this is a 50 ml conical tube. I don't think I showed this before. So this holds 50 milliliters of water of liquid. I have water in here, and this is molecular grade water. So basically, it doesn't have anything that can break down DNA or RNA. We're not working with RNA, but if I was, I wouldn't have to worry about any kind of enzyme showing up in here that can break it down. And those are usually called DNase or RNase. If you hear that, that's what that means. If you hear about a DNase, that means it will degrade your DNA. If you hear RNase, it will degrade your RNA. And we don't want that. We want to keep our plasmid DNA intact. Plasmid DNA intact. All right. So we're going to do a minute. We just want to make sure. I know some of these have sat longer than a minute. We're just being thorough with it. They have to sit for at least a minute. I'm going to put this back. I don't need this. And then we'll do one last centrifuge of these guys. So one more spin. Same thing. A minute at 18,000 times G. It will pull the water through our membrane, taking the DNA with it. And then we'll set up our concentration reader. So we're going to use a machine called a qubit, and what it does is we're going to add a dye that will bind onto our DNA, and it will fluoresce in the machine, and the amount of fluorescence that is in our sample will tell us how much DNA we actually got. So it's a nice little machine that tells us that we have DNA. All right, 1 minute. I'm gonna set these in the centrifuge. Get these guys going. All right, 1 minute. This will be all set, and I do need to set my timer again for 2 minutes, and that is required for the concentration, for us to read the concentration, because you need it to sit for 2 minutes to let the dye actually sit on the double-stranded DNA, so that's why I'm putting it there. And then we'll be all set. I'm excited to see how much DNA we were able to extract from our samples. Ooh. And I will need these guys. So we will take that 1 microliter of our sample. We only need 1 microliter. We don't need any more in order for us to measure the concentration, which is great. So we're not wasting all of all of our samples, just measuring how much we have. So that's nice. And then I just want to make some more room on here. There we go. Get myself all set up, prep and ready to go. And then, so essentially this last spin, that is the end of a plasmid prep. That's the end of an actual plasmid extraction. So that protocol's done. That's done with the kit. We're all set. This next step is just what I like to do to see how much I get and how much I'm going to be working with when I do the enzyme digestion. So the restriction and the enzyme digestion, that's going to happen next week on stream. All right. Samples are ready, samples are done. I'm also going to turn off the centrifuge since we don't need it anymore. And it's kind of loud. It's loud background noise in here. So we're all set. Get rid of the column. And there we go. There's our water down at the bottom with our DNA. So guess what? We just extracted DNA together. Isn't that amazing? You can't see it. It's too small. It's too small to be able to see. But it's there, and we'll be able to tell based on what the equipment tells me. There. All right. There we go. DNA extracted. I'm gonna move these in the order they should be, just so I don't mix up while I'm doing the concentration. All right. So 1 through 8 of C and D. I'm going to get what we need in order to do the actual concentration measurement. Just give me 1 second. All right. Let's see. So here we go. Sorry, I moved my microphone. It seemed like it got louder because I moved my microphone back. So the instrument requires these special tubes that are very clear. And here I can show you, compared to the microcentrifuge tubes, these are extremely clear. And the reason is because we're going to be looking for fluorescence. It's going to be looking for an emission. So something that's going to light up. And it has to be clear, very clear, in order for it to be able to see in the instrument. Oh, you know what? Make my life easier. So we have 8 samples, and I'm going to be using the same buffer. So it's a qubit double-stranded DNA buffer. What does that mean? It means it only will look at the concentration of double-stranded DNA. They have different buffers for single-stranded DNA and RNA. So this is specific for double-stranded DNA. But because I have 8 samples, I'm going to make a master mix. A master mix is basically you're using the same buffer, the same components for all of your samples. It's better to just measure it all out in one big tube and then put it in separate tubes. It's less tip waste. It's less effort, essentially. It'll save time. And so each of these, I gotta do some math. I'm pulling out my calculator. Oh, TI-83. Oh, I've had this for a very long time. I need these 8, so we do 199 microliters for each of the tubes of buffer times 8. So about 1.592 mils, and I'm going to divide that by 2 because I can't pipette that volume with my micropipette. But if you divide that volume by 2, it is 796 microliters. And grab my tips, and that's what I'm going to put in here. So here's one. Here's two. It's right at the top. Right at the top of that tube. If it was more than that, I would have used a different tube. Because we're like, skirting the edge right there. I don't need this anymore. So the other component that I need for measuring the concentration is the dye. So this dye, like I said, is specific to double-stranded DNA. And we have 8 samples, so I need 8 microliters, because you use 1 microliter per sample. And I'm going to put that directly into the master mix. So if you can see it, you might be able to see it. It's a red color. So there you go. You can probably see it really well. So it's a red color. And we put it right into the buffer. Close this up. We have the aluminum foil over it because this is sensitive to light. It'll actually start breaking down in light if you leave it out too long. So we have it covered in aluminum foil to just help that it doesn't do that. Then I'm going to mix this up. Just like that. A couple times. Just to get the dye to go throughout the buffer. And then what I'll do is aliquot, which basically means I'm going to pipette the amount that we need for each of the samples from the master mix. So I'm actually going to take the top, because we want to have as much as we can. And then I'm going to put it in each of these tubes. So it makes it a lot easier when you're dealing with... I usually make master mixes if I have 3 or more samples. Because that's just the limit I set for myself. 3 or more, make a master mix. It's just easier. It doesn't waste time. And doesn't waste as many plastics as well. So I have 3 or more samples, master mix. All right. Okay, so we're all set on that. I'm going to put this away. Get that out of there. So now I'm going to actually go put the buffer and the dye away so they're not sitting out anymore. And like I said, keep a clean bench to work on. Then I won't have to worry about knocking it over as well. Because sometimes you get those clumsy times where just flail and knock stuff over. All right, so we're going to take a microliter of each of our samples and put it into the tube. Now, if you wanted to label these tubes, you can. You just can't label the sides because the instrument will take the measurement from the side of the tube, and if you label it, it's going to change how the instrument reads it, because if you have black or blue and it's reading through the tube, it'll actually change, it'll make it look like your concentration is lower or higher than it actually is because you're getting the light hitting something else. So if you wanted to label the tube, you would label the top. And then I'm just tapping it to mix. Make sure the DNA is not sitting at the bottom of the tube. I want it to be a mix so I get a more accurate concentration. And I am pipetting the 1 microliter directly into the buffer just to make it easy, and it's common practice. Because if you put it on the side and you forget to mix it or forget to spin it down real quick, then you're not going to have any of your sample where you want it to be. So it's just good practice to pipette into the buffer. There you go. Now I'm not labeling the tubes because my system is 1 to 1, 1 to 2. Right? Like 2 is there, 3 is there, 4 is there. So I'm not labeling it. I know. Just because I know I'm not going to mix it up. I'm not going to mix it up. And we don't keep these tubes once we've read the concentration. There's no reason to, and plus we can't take our sample back. So that's why I'm only using 1 microliter. We can't get that microliter back. We just end up throwing it away once we're done. But everything else, we have our original samples. But what I'll do is when I go over to the instrument, and I won't be able to show you the instrument just because, like I said, this is a fixed camera. I will write the concentration on the side of the microcentrifuge tubes and in my lab notebook. So I have it in two places, and if I just want to grab a tube, I don't have to refer back to my lab notebook every time. It's right there and easy to see. All right, so that is set. Put this back. Then we just mix this real quick on a vortexer. So a vortexer just basically mixes really quick, really fast. And that's to make sure that the sample and the dye contact each other and that DNA is everywhere within the buffer. And then we let this sit for 2 minutes. And then I'll go measure the concentration, and I'll let you guys hang out with the music while I do that. Since it's not gonna be fun. I'm just gonna be over at the instrument in that case. So I'll let you guys hang out with the music and enjoy that. But yeah, so yay! I'm so glad I got to show you how we actually get DNA from an E. coli cell, and we basically have gone a little bit full circle. We started off with making double-stranded DNA, our plasmids, then we let them grow up in E. coli, and now we're back to trying to get our plasmid that we made. So cloning, this is cloning, and it's basically done, although you do obviously want to check and make sure that what you made is correct. But say if you just wanted to make more of a plasmid that you already know is correct, that you've already done before, that's called subcloning. It basically means that you're just growing up more of something you already know that you have. You've already checked it, you sequenced it, it looks good, your stock is just low, and you need to make more of it. So you just subclone it into E. coli, do the exact same process that we just did, but you just don't have to check it with restriction enzymes if you've already done that check. And sorry, this is my pen. If you're like, what is that weird thing? It's my marker so that when I go check the concentration, I can write it on the side of the tubes. But yeah, so cloning, pretty awesome, isn't it? I know, I like it. When it works. Sometimes troubleshooting it is not fun. It's not fun. So like I said, A and B, we weren't able to get anything from that. That's fine. I will have to troubleshoot that, mix it up a little bit as far as moving forward, but hopefully C&D is going to look good. We'll find out next week. All right, so it's been 2 minutes. I'm going to go check these concentrations, and then I'll be right back. All right, I'm back, and I measured the concentration. It was relatively quick, but, you know, just in case. So they all looked really good. So the lowest I think I got was 270 nanograms per microliter. What does that mean? that means in 1 microliter, we have 270 nanograms. So it's a small amount. So if you think of gram, you've heard of a gram before. We're talking very small, very small. So you can't obviously see it. But that's a good concentration. That is very good. I went all the way up to, I think the highest concentration is 1019 nanograms per microliter. So that, once you hit 1,000, it becomes a microgram. So we go from nano to micro, which is good. That's a lot. That's a lot of DNA in 1 microliter. So plenty to use for downstream protocols, which is great. A bad concentration is anything that would be like 50 nanograms per microliter and below. That usually means that it didn't grow well. It's not enough to really check anything. You need to have a good amount of DNA for the restriction enzymes to work so you can be able to see it on a gel. So if I got 50 nanograms or below that, I would be sad, for one. And I wouldn't be able to use it. I wouldn't be able to do anything with it. It's just not enough DNA to work with for what I have planned as far as the restriction enzymes go. But yeah, so awesome. This is great. I'm so excited. Plenty to work with. I'm actually going to go put these in the freezer so they will stay more stable. And I will be right back again. All right. Well, the protocol is done for today. I'm going to take off my PPE. Don't need my lab coat right now. All set, I'm going to wash my hands. And I can do a recap of what we did today. See if anybody has any questions in chat. And then I can give you a little drawing preview, I guess, of the restriction enzyme digest that I've been talking about and what I plan on doing next week. Just so you have an idea of what I'm talking about in that case. And I don't know, maybe the drawing will help. But I'm going to grab my markers and things. There we go. Sorry, I was just checking something. But yes, how is everyone doing? I hope you're enjoying the stream still. Vibing. I'm gonna put on new gloves just briefly so I can clean up my bench. Ugh. Of course I washed my hands though now. Now my gloves don't wanna get on. Gotta love it. It's stuck. It'll work. It works. Alright, I'm just gonna clean up with some 70% ethanol. So the bench is nice and clean. Let it dry off before I bring back our mascots. I don't want them to get all dirty. No, we don't need some really coli on Eli Ecoli. That would be bad. I don't want to contaminate our mascots. There we go. But here we go. Here's our mascots back. So, yeah, so brief recap of what we did today. We extracted double-stranded DNA, plasmid DNA, from Eli Ecoli. Just checked the concentration, so how much DNA we actually got. Looks great. I'm very happy with the concentrations that we got. So that's awesome. So we basically, poor guy, we punctured holes in his membrane and let the DNA come out along with the rest of his. Whoops. There we go. Sorry. Had a brief little drop some frames. The frames kind of went bad on me for that. Yeah, so punctured holes in his little membrane, got the DNA out as well as the rest of his cellular components came out. So we were mean to Eli today. Oh goodness. Getting some lag. Is it better now? Sorry for the lag. Sorry for the lag. There we go. I think it's better. I think we're back. Yeah, there we go. Apologize for the lag. Don't love the lag. We need a lag monster. Oh no, it's still happening. Let's see if it'll clear up here. Let's just wait, see if it'll clear up a little bit. Definitely lagging right now. Network said no. Give it some time. Ooh, still laggy. Just wait for it. Wait for it. Gosh, I hope I don't have to end stream because of the lag. Is it getting any better? Oh, I don't think it's getting any better. that help any? Is it any better? Any better? It's just dying on me. Minor issues. Okay. Might be coming back. Might be starting to come back. Nope. Well, I had to turn off closed caption. Okay, everyone. I don't know what is happening. We're doing so well. I'm gonna have to end it here, I think. Maybe. Maybe not. No. It's just getting bad. Alright, the lag isn't going away. Alright, thank you for coming in my stream. Hopefully you can still hear me. I think the lag has just knocked out everything. I appreciate everyone in chat. I appreciate the lurkers as always. If you can't hear me, I will put in chat, but it looks like the lag is just taking up my stream. But thank you again.